Thanks, PacBio, for inviting me to be here. And we just discussed that in the break before, that I was here for another PacBio user meeting, I think in 2019 when SQL 2 was launched. And oh boy, how far have we all come? I mean, it's amazing. So I'll touch largely on the rare disease research, but also really the push towards clinical applications. And if I stand here, I certainly stand here on behalf of a great team of many people and many amazing colleagues in Nijmegen. If I sound excited, I hope you believe me. It's not only because PacBio sponsored a few reagents for some of the projects, not even all. The real reason I'm here is, of course, the ongoing long-read revolution. Maybe I should even update this paper to the Methods of the Year paper. But I think since a few years, we really saw the potential here. And I think especially with Revio, that has matured enough. The scalability is starting to really make a push towards clinical applications and for humans. So we, different from people that work on metagenomics, our damn genome is pretty large. It has a lot of repeats, so we really need higher throughput. And with Revio, we're moving into the right direction, of course. This is our facility. We invested quite heavily in long-read technologies over the years. And I particularly wanted to use this slide, first of all, to show you our email address, but also to introduce Janneke and Ronnie, who are also here with me. So if you have questions or want to collaborate with us or engage with our facility, those are the two to also talk to. Of course, that picture didn't contain the Revio. Our first Revio was installed in April 2023. We were so excited that we directly got a second one installed end of last year. And we also engaged in one of the collaborations with PacBio. And I'll really showcase some of the work out of these collaborations. And it's in essence answering questions that are quite similar to the questions we ask in this HiFi Solves EMEA consortium. But then basically the local version of that. And if you think about pushing a technology towards clinical applications, there's basically two questions. And they're relatively simple. The first and obvious question is, could the new technology, in this case HiFi genomes, potentially replace all of our germline testing? And I'll really allude to that, why other technologies have simply not managed to do so. And we've tested that question by taking 100 incredibly hard to identify mutations. In that sense, you could say it's boring. It's like looking at cases where we already know the outcome. But I think technically speaking, and more from a diagnostics QC kind of point of view, those are incredibly important. But those are very, very hard mutations, and I'll certainly spend some time on those. But then the second question is similar to what Dave already mentioned is, and that potentially is clinically and scientifically even more exciting, how many more cases can we solve? And that also means how many more families and patients can we ultimately help? And we there have different flavors of that, and I briefly touch on that as well. And then I have a more representative study that answers a similar question. As David already mentioned, we're engaged in this HiFi Solves EMEA consortium, so I have 2 or 3 slides on that. And we're currently preparing a much larger prospective study that really would be the ultimate push towards clinical application. But just to kick off with a little bit of fun, of course, we're all a little bit competitive, but we're all friends here, so I want to invite you to use Twitter or X, despite all the recent kind of issues with it. At least to post your most successful runs. And there's Ronnie and his amazing team. They're crunching out quite incredible libraries at the very moment, largely prepped manually. We're optimizing the automation of it. But our latest record is 127 gigs, so that's a 40x genome. So that actually starts, let us start thinking whether we could even pool 2 genomes on a smart cell soon. And that obviously would like double the throughput. So that would be quite fun. So you're all invited, tweet your records and beat us. I mean, we all have to learn from each other, obviously. So for the rest of the talk, I really focus on rare disease, and I thought at least I'll bring one overview slides for those that don't work on human genetics, but other genomic applications. And even if you work on human genetics, maybe you don't work on rare diseases, and hopefully convey the message why rare diseases are so important. The name just says it, rare diseases are rare. Some of them are ultra, ultra rare. There's sometimes only tens of cases globally. But there's so many rare diseases that collectively, they're incredibly common. They're even that common that 1 in 16 or 17 individuals has to do with a rare disease throughout their lifetime. And it's thought that 75 or 80% of rare diseases are actually genetic, and quite often monogenic or Mendelian even in its inheritance. A lot of the rare diseases manifest in childhood already, so quite a lot of those are really inborn issues, if you wish. A a lot of those diseases are extremely very severe, so a lot of children suffer really badly from it or even pass away early in their life. And the big trouble is, while we have improved by exome and genome sequencing to diagnose rare disease cases, there's still more than half of all patients that simply don't get an answer from us, and they're terribly disappointed. So we really still fail for the majority of patients, and that's where our duty comes in to innovate and keep on pushing the knowledge, but also keep on pushing the technology to do better. And this may be a bit of a simplified slide how current genetics is run, or how in the past genetics was run. I think we have already improved over the last decade by NGS. So quite often, there's an entire so-called diagnostic odyssey happening, and patients are seeing multiple doctors throughout their lifetime, and multiple even genetic tests are done throughout their lifetime, until after many, many years, they still don't have an answer. So this kind of cascade testing is really what genetics was all about. And it all started with karyotyping and all those bloody technologies that only delivered an answer in a very small percentage. I think nowadays we really have exome or short-read genome sequencing as one of the first-tier tests, so that looks much better. And we also push those tests much earlier to a clinical trajectory of a patient. So in fact, instead of seeing 5 different clinicians, you just need one clinician that decides this could quite well be a rare genetic or Mendelian disease. Hence, we would sequence very early in the process, and then you see the patient much more detailed once that genetic test is available. I think that's the more ideal scenario. And if we talk about that, we already know the first-tier test of exome sequencing is already cost-effective. However, genomes would, of course, be much nicer. Exomes, by definition, are not complete. I think they're the best first-tier test we ever had so far. Genomes should be better, but we'll dive a little bit into how the current genomes are actually for that purpose. And I just want to highlight on 1 or 2 slides a study that was led by my dear colleague Lysenka Fissers together with our huge diagnostic unit. So what Lysenka and her team did, they went back to 1 year of diagnostic production. And that is, we run more than 30,000 tests in our diagnostic unit. And you see the individual test modalities on the bottom and the number of tests run per test. So that's just, it's an entire test portfolio where exomes and by now genome sequencing are really taking over. But the big question is, can they replace all of that? And the sad news, by running representative samples for 1,000 genomes now from all these different test portfolios, the sad answer is no. Illumina or short-read genomes cannot replace all of those tests. They're doing quite well to replace exomes, but then there's a lot of other tests where you could potentially diminish the number of tests, but you can't fully replace the workflow. And that's another part of the equation in a diagnostic lab. Ideally, you just reduce the total number of workflows dramatically because you still need to train people, you need to pay maintenance for the machines, you need extra lab space for some machines. So actually, short-read genomes, that's the short answer for that study, are not the final answer. It's not the generic test that we can offer in the clinic. It's probably a very good first-year test, better than most of the other tests we did, but it certainly won't replace the majority of tests. And if we look at that in a slightly different way, it's only one part of the pie that we're really missing, but that is 25%. So a quarter of patients that wouldn't get the current diagnostic answers if we would only do short-read genomes. And on top, as I said earlier, more than half of our patients don't get an answer from the current test portfolio at all. So those are the 2 questions that we need to tackle to do much, much better. So that brings me really to the first question that I briefly introduced. Can HiFi genomes potentially replace all of the germline testing? I think it's not fair yet at this stage to talk about somatic mutation testing in cancer or leukemia. I think that naturally would come with much higher depth of sequencing. So I think we're not really looking for HiFi genomes for that application right now. I think in the future that may well be possible, but certainly their scalability is still a few more years down the road potentially. So we took 100 mutations where Illumina sequencing from the previous study really failed. So from all different test portfolios where we already know that they're really hard to tackle. And I go back to that pie chart, we left out the somatic mutations because there I think it wouldn't be a fair assessment. So just to give you the details, those 100 samples are collected in a way that we look at the very, very tricky mutations. So some are mutations in homologous sequences, partially in pseudogenes or genes that have pseudogenes to be precise. You'll see some of these very difficult genes on the upper right of the slide. We looked at repeat expansion disorders, 14 different genes. So those are two classes of variants where HiFi should do much better than the standard of care, hopefully. Then we had some other homology regions, pseudoautosomal regions, where we had mutations. We have some indels and homopolymers. I think a lot of other technologies have severe issues with that. We now can already say that HiFi is really good at it. Then there's several complex structural aberrations. There's some imprinting or other methylation defects included, and a bunch of other samples where there was a slightly different diagnostic question. So that really represents a good average of what our diagnostic lab has to deliver. But then only the toughest of those mutations, keep that in mind. So I'll just give you some snapshots of what was included. Here we had a relative straightforward 3kb deletion, but in an extremely GC-rich region. See, it's involving exon 1 and the promoter. So there you almost have no coverage in short-read sequencing. That's why why Illumina can miss it. But here it's very, very clearly present in the HiFi data. And a nice add-on, and we really still need to get used to that, you directly phase. I mean, for now we talk about variants, but in fact now we can start talking about alleles and haplotypes. So proper mind shift for the entire field, and certainly much closer to reality. Then we had more tricky structure variants, mobile element insertions, totally nightmarish to tackle with short reads. We had several of the homologous sequences using Mike's team's tool, the paraphase tool that is now available for, I think, 160 or so loci. So several of those are covered. I'll give one more example later from HiFi Solves. Several repeat expansions are in there, and then methylation defects. So these are just 5 of the mutation-positive cases where previous tests really had a big trouble in finding them. But to summarize that really in statistics, so far we have more than 90% success rate. And there's really just 2 issues that we see a bit more systematically. One is for some of the very long repeat expansions, we just don't generate enough coverage and we miss those mutations. And we can discuss why that may be, or I'll actually give that away in a second. And we miss 2 very complex cytogenetic aberrations. One being a Radboudumc translocation. That's where the acrocentric P arms break and fuse. I mean, to be honest, any kind of sequencing currently would have major issues in finding those, so probably really put the technology at a test here. And then there was another translocation that also had breakpoints in very, very complex, repeat-rich regions of the human genome. So in that sense, none of that was a proper surprise. I would rather say it's a surprise that we really find 90% already straight away without any fancy reanalyses and stuff. I think by now, people start understanding what the issue may be for some of these repeats. It's in particular RFC1, and another GAA-rich repeat for Friedreich's ataxia. And we tend to believe that may be a secondary structure formation on the DNA. And Alex Salazar and his team have discovered that and have published part of that. And I guess he will talk about that in his talk. For the Radboudumc translocation or other typical cytogenetic aberrations, there may be a chance to utilize the telomere-to-telomere reference genome or perform local assemblies or so. None of that has happened yet for that study. So even there is hope to tackle some of those. Maybe we may not manage to tackle all of them. And just to give you one of the examples for the repeat expansions, the initial study was run on the old software setting. So we simply, for the Friedreich's ataxia case, that was a failed one on the previous slide. There you see it. Diagnostic said it was more than 65 copies on each allele, so therefore disease-causing. But we simply had no reads whatsoever. And only since the more recent software upgrade, we repeated the sample, and now we get data, we get coverage, and that is by basically recycling or reusing the failed HiFi reads. So those don't reach HiFi quality, but are still very useful reads, and now you can recycle them and use them basically. Some call them the bonus reads, and it's actually quite a lot of bonus reads. And now we can even tell that we outperform current diagnostic testing because the standard of care tests are not great in determining the actual repeat length. Now we see one allele has 309 repeat units, and the second one has 328. So that is quite nice. So there is recovery for some just by the software upgrade and utilizing the failed HiFi reads. There's one other workaround potentially. If you see drop of alleles, that already may tell you there is a deletion or something else present, which would make sense, so that you know already that there would be something suspicious at the locus. But as Dave already introduced, there is a new targeted approach where you can use much, much deeper coverage. So for some loci, this certainly should recover more and more of the reads. And we were on an early access for this cool pure target assay, and we actually quite like it. Our diagnostic expert for repeat expansion disorder said, wow, that's the assay I dreamt of for the last 20 years, and it's finally there. So pretty cool. Just to give, I think, 2 or 3 slides for the HiFi Solves EMEA consortium, so as Dave kindly introduced, that's a bunch of really highly respected colleagues around Europe that all have kind of a common goal, that they're really well established in a clinical setting and really want to push this technology towards clinics and clinical applications. And for the first tier, we wanted to dive deeper into the homology challenge. So looking at mutations in genes that have pseudogenes or otherwise homology. One group in particular is from Biosensia, so Christian and Hanno in Germany. And just to give you one first case from their group, so they looked at a small 18 base pair deletion in one of Hanno's favorite genes. And you see in the upper panel, you see exome Illumina data, and they just look terrible there, obviously. So that's a very, very complex region of the genome, so it's not reliable at all to do that with short reads, let alone to be able to call an 80 base pair deletion in the little red box there. But with the HiFi data, you not only find the variant and the mutation very clearly, you also get phasing information. So that already shows what a leap forward the new technology is. One of our own examples is one of these genes that has a lot of homology issues, stereocylin. Here we had a hemizygous point mutation, a stop mutation even. So if you just look at the standard HiFi data, you see the upper panel. That's not quite exactly easy to use yet, but then you have the new tool called Paraphase, and I just show you a little zoom in for that. So that gives you automatic haplotyping. So the upper rates map to the stereocylin locus, haplotype 1, but the lower 2 ones just map to the pseudogene. And then it's beautiful. You can just zoom into that allele that is there. So first of all, we see that one allele is deleted. That's what we knew from the previous test. And the other one has this hemizygous stop mutation. And you can just zoom in and directly have the phasing information. I think previous technologies never delivered that. So that's really, really exciting. Right. Sorry for that. So that was question 1. How good can we do for terribly difficult but known mutations? The other question, obviously, is how much can we mean for patients where we don't have an answer yet? So if we do that for unsolved rare disease cases, we and several others have kind of pioneered patient-parent trio sequencing because de novo mutations are so important. In particular, for severe sporadic diseases, they're probably the most important mutation type. Good news is they're extremely rare. Bad news is you need a very accurate technology if you look at that from a technology perspective, because any false positive you make in the child would look like a de novo mutation. So if your technology is not up to high standards, you can't do that assay. So therefore, we always like to include trios quite early on in a technology, and we even did so for Sequel 2, and I'll show you the data on the next slide. So if you now go back from just a technical perspective of de novo mutation to the real biology, we know about point mutations that every one of us is born with 50 to 100 point mutations across the genome. However, the de novo mutation rates for indels or STRs is much less understood. For structure variants, the same is true. We simply don't know really well what the de novo mutation rate is, simply because technology wasn't mature enough to say so. But it is anticipated that de novo structure variants are much more rare and that mutation rates are much below point mutation rates. So it's anticipated that in a human genome, we all carry between 0 and 2 or so, that is de novo per generation. And if that is true, my prediction is that in particular de novo structure variants will be the ones that crack the undiagnosed, unsolved cases. And scientifically, those are the exciting ones that will teach us new paradigms. So those will tell us what happens in the 98% of the genome that we don't understand at all, the non-coding regulatory space. So my prediction is that structure variants in the non-coding space are the ones that really teach us very important lessons, and therefore they're overly exciting. Not easy to understand, I admit that, but they're really exciting scientifically. Here's just a summary table of our first 8 HiFi trios that we did with high coverage. So that is Sequel 2 data, but 30x coverage. These are the numbers of variants you find in each and every genome. On average, 4 million SNVs, 1 million indels, and close to 50,000 structure variants if you start counting at 20 base pairs. It's more in the range of 25 to 27,000 if you start counting at 50 base pairs. And then below you have the de novo mutation candidates from that study. So we reproduce quite well what is known for point mutations. We add a few indels that are quite unique then to the long-read data, but certainly the structure variants, the vast majority of those across the 8 trios have been missed previously by short-read data. If you want to read up, I don't bore you with all the details, that paper just came out last year. We embarked on 8 trios. The sad news of that story, and you can read it up in the, you can see our depression in the discussion a little bit. We didn't crack a single case. And imagine 8 of those trios were quite expensive on sequel 2, obviously. So kind of a disappointment from a more clinical and discovery perspective. So therefore, now we embark on 100 trios in our tier 2 clinical utility study. And I'll just present the first 2 examples there. Obviously, in such a study, you start finding de novo mutations that are potentially clinically meaningful. Here's just one example. We filtered again for all the de novo structural variants, and we immediately found 4 de novo break-end calls. So what does that mean? I'll just illustrate that. It's a patient with a quite severe neurodevelopmental phenotype and some syndromic features. What we really saw in this patient is that there was a more complex insertion-deletion on chromosome 2. That means we had a deletion on chromosome 2 of 19 kilobases, so quite large. But there was material inserted that was duplicated from chromosome 3 into that deleted part of chromosome 2, so a more complex structure variant. You could then always ask, okay, why was it initially missed? Initially, the standard of care test was just an exome. And here you see the deletion that could have potentially been called, but at that stage, we only used conifer in the clinic, and only 3 probes showed a deletion, so it was really a borderline call. And the gain that was actually inserted, obviously that insertion is not visible at all in the exome sequencing data, but also even the gain was only seen in a single probe of the exome data, so therefore was completely missed. So what we think is going on here, we have this unbalanced translocation or deletion insertion event between chromosomes 2 and 3. And my best guess for now is that this CADPS gene is still intact in 2 copies, but the insertion of that material in ADAM23 may have disrupted one of the ADAM23 copies. It's not a known and established disease gene, so potentially a novel candidate gene to follow up. And the constraint against variation in that gene is striking. So I would be interested if anybody has ever found other de novo mutations in that gene. Maybe we should compare the phenotypes of our patients, and maybe that leads to new discoveries in the future. Just a brief second case example from that clinical utility study of so far undiagnosed cases. Here we found a much simpler structure variant, a single exon deletion in FOXP1, which in hindsight fits the phenotype extremely well. So I think this is very close once confirmed in making really a diagnosis for such a case. And that was simply missed because it only touches 1 exon, and that was also not called in the previous test. So those are maybe just anecdotal from the first data. The rest of the data analysis and importantly interpretation is currently ongoing under high speed. But I brought another study that is much more representative in a sense to really give you an answer how much extra can we find. And that comes from our SolvRD consortium. Some of you may have heard about it. That is a pan-European project involving 300 people across the entire continent, amazing partners, true disease experts for various rare disease classes. And what we did here, we met at one of our annual meetings. What we did there specifically was also to sequence close to 500 HiFi genomes. However, that project was conceived several years ago, so at that stage we only afforded 1 single smart cell on Sequel 2E. And we've now finished sequencing and finished sequencing of 480 samples, analysis of which, but interpretation is still ongoing. But I already have the first data freeze, close to 300 samples representing 114 families from this project. And just a month ago, we had a specific long-read solvathon in Nijmegen, so really brought together the clinical disease experts, some of the genomics and bioinformatic experts, and really hang out for 2 days in a room, similar to a hackathon that was organized in Sweden last year. And that's so much fun, and it's also very productive. There's cases you really solve on the spot if you just bring the right people together. So I can strongly encourage to do that, and I'll just highlight on 2 or 3 slides what we found in this cohort. So there were 2 cases included that had muscular dystrophies, but no mutation found by the standard of care methods. Several even underwent a lot of previous tests, and we found 2 inversions breaking the DMD gene. So that was immediately jackpot. In one of the cases, it even breaks a second gene. I think, let me see, that's the the lower case, it also breaks NHS. So actually, that patient suffers from a phenotype. It's two Mendelian disease genes, DMD and NHS. And actually, in retrospect, that disease also fits a merged phenotype of the two, where DMD clearly is the dominant case in the clinical setting. Just to highlight a few others, we also had diagnostic value and validated variants in 4 other cases, all of which were repeat expansions. We only had one single smart cell, so only 10-fold on average HiFi sequencing, but that can be enough. I mean, the data is so good that sometimes just with 2 or 3 reads supporting a long repeat expansion, you're clearly there. And some of those phenotypes are recognizable. For some, we even have orthogonal validations or RNA sequencing on top that really supports that we cracked those cases. So if I sum up next to these examples, what we found across the entire Solv4D project so far, we really solved previously unsolved cases in 11%. And excitingly, and so far it's a very much low-hanging fruit, conservative estimate, 6.5% of extra candidates, some of which are currently undergoing validation or segregation to make the point. And I think that's really just the first grasp of the data. Some of the clinicians or clinical scientists involved really just because of the Solv4D got so engaged that they're still studying several of their candidates, and they're not even mentioned there. So to sum up the Solv4D lessons, even with relatively modest HiFi coverage, we find disease-causing mutations on all levels of variation. So point mutations, SNVs and indels, STR repeat expansions, but clearly also some structural variants that had been previously missed. So yield there is close to 11% and 6% or so extra candidates that we still follow up. And that's actually huge. Those are the hardest to crack rare disease cases from all across Europe. So any extra gain of a single % is already so important because it represents many family members sometimes, and families that went undiagnosed for sometimes a decade or more. What I should also admit is we had some of these unsolvable syndromes in there that you may have seen and spotted some of the previous slides. There's some syndromes that are clinically extremely well recognizable by well-trained clinicians, including Hallermann-Streiff syndrome, Gomez-Lopez-Hernandez, ICARD, and a few others. And the sad news is none of those unsolvables was solved. Not a single case, nor did we find a common case for any of those. So there's something really tricky, but maybe they really give us new paradigm insights in the future. So several of those will now be repeated on HiFi Revio sequencing with higher coverage, looking at the methylation in parallel, sequencing yet another tissue, sometimes the affected tissue. Maybe we crack some of those. Maybe some of those are not genetic after all. That may well be. Maybe we as geneticists overestimate in some cases how important genetics is. Okay, I'll just finish by telling you the why. Why are we motivated to do all of this work? In one of the much more impressive rare disease cases where innovation and long-read technologies really made a difference. And this is a collaboration with my previous colleagues in Nijmegen, Marjolein Jongmans and Roland Kuiper, who moved to Utrecht. And they were seeing a very impressive Dutch family that witnessed full tragedy, if you wish, because they lost 2 of their daughters very early in life due to a terrible brain tumor. And if this happens twice, we know quite exactly what's going on. It's a very specific type, ATRT, an atypical teratiary rhabdoid tumor. If you see that in 2 cases in a family, you should have a very strong suspicion for a germline predisposing mutation on one specific locus, namely SMARCB1. And they did histology in one of the brain tumors of one of the daughters and even confirmed that in the tumor, SMARCB1 was completely out. So that must mean there is a germline predisposing mutation and a second hit happening in the cancer quite early on in development. So with that suspicion, they underwent the entire diagnostic portfolio. Sanger sequencing was done, MLPA was done, CNV microarrays was done, exome sequencing, all in a clinical setting. And despite knowing where exactly to look, you couldn't find the mutation. Then Roland's team in Utrecht took up Illumina sequencing, genome sequencing, and still the mutation was missed, while knowing where to look. Not a surprise, the mutation still is in the so long suspected locus, and only because of our long-read technologies we found it. So in this case, it was early days. We only had the sequel 2 going. We used a combination of optical genome mapping and PacBio HiFi sequencing, and now we understand what the hidden mutation was. It was a retrotransposon insertion in intron 2. So it's a specific super nasty retrotransposon, an SVAE, has a super long poly A tail, and it has ALU-like elements, VNTR elements in the middle, so extremely repeat-rich. No wonder short reads can't tackle those kind of events. Roland's team has evidence now on RNA level that really disrupts one of the allele in already germline. And if you're interested in more details, read up. It's published 2 years ago. But maybe even more motivating is that I had the opportunity to personally meet the family that was involved. And I'm super grateful that we work in such a translational clinical setup, because what we did is, after knowing finally the disease-causing mutation and the mutated allele, my colleagues in diagnostics dared to offer immediately, or develop and offer, a prenatal test. Luckily, the mother of that family was only 40 years of age, and it worked out. She got pregnant again. And very early on in the next pregnancy, we could offer a prenatal test. And lucky circumstance, there was no mutation present in that pregnancy. And now, since 1.5 years, they have a healthy baby boy. And I had the true privilege to meet the family for a full day, and we even took a little video of that. A truly amazing family who really just deserved this answer. And I think we really changed their life for the better after the full tragedy that they witnessed. And that, honestly, is a much bigger motivation than some of the papers we publish. So I think it's time to wrap up. I hoped I conveyed some message how cool long-read genome sequencing is. I think it's fair to say that 30x HiFi genomes are extremely comprehensive genomes. I think we're even enabled to find de novo mutations on all levels. I think better than any technology ever before. One specific emphasis is that in particular structure variants, and that's not new, Evan Eichler is praying that for 10 years now, especially structure variants and other more complex events are only identified by long-read technologies and are clearly missed and hidden from short reads. We, in Nijmegen, but I think also several other partners globally, believe that there is a huge potential for HiFi sequencing as a proper first-tier generic test for germline testing. And that's the main reason why we run all these studies that I tried to present today. HiFi Solves as one of the representative rare disease projects, but also Gregory in the US is Gregory is I think even global, and similar studies are now ongoing that all show that doing long reads in previously undiagnosed cases after exome and genome sequencing, you can really crack 10% quite easily. Probably there's even much more in the data that we don't fully understand. I think it's a proper mind shift that is ongoing in our field. So far, due to technical limitations, we had to talk about variants. But variants obviously don't live in isolation. They all happen on the backbone of our genome. So we actually should talk about alleles and disease haplotypes. And I think due to the limitations, we never did. I think there's real promise from some of the studies we do locally. And it's beautiful to see, for example, in HiFi Solves, that we're exactly aligned there and that we're replicating all the clinical utility there. So with that, I'll finish and just invite you to jump on the long-read revolution. And I think we only got started, so I think the next 10 years or so will be overly exciting. So I think it will be an amazing journey, and we only got started on that one. With that, I'll finish to thank a lot of people, several people from PacBio, potentially many more than there on the slide. Thanks for the journey. Thanks for the proper collaboration. I think we're much more than just a customer, and we thoroughly enjoy that. So thanks to all of you for your hard work. Bring that back home to the R&D and other labs in your institution. Many thanks to all the partners in HiFi Solves EMEA. Several of those are here, of course, and talk to them. Solv4D involved more than 300 people across Europe. Here only those mentioned that really contributed to the long-read paper of Solv4D. Then many people in Nijmegen itself, and I'll finish by highlighting again our facility led by Janneke. Ronnie is here, as I mentioned, so approach them if you have really nasty questions. I can only answer the easy ones. And of course, my very own research group. And with that, I'm happy to take questions now or later in the panel, and I'm around for the entire event if you have questions later. Thanks a lot.